Hi everyone, apologies for last week. I had a medical emergency at like 5:45 and I completely forgot to let everybody know. So by now we're getting into the nitty-gritty of organic chem. Fun part of learning everything is done. You should be starting on amino acids, proteins. Some of your teachers will take you through polymerization first. I like to do it this way, just the way I do things. Now we're getting into what you call biological chemistry. Very creative name. Now we're looking at, today we'll be looking at amino acids, proteins. I'll then create another session and then we'll do action of enzymes. Week after will be carbohydrates. Then we'll start looking at plastics and we'll round out the organic chem stuff, and then we'll spend a couple of weeks just smashing out practice exam questions. I'll also be running this next term as well. So for industrial chemistry, which will lead us into term 4, and then term 4 is mostly about revision. Okay, let's get this going. So proteins, everybody remember what a catalyst is? Remember back to our equilibrium unit. So catalyst being reduces the activation energy required for a reaction. Protein can also be a hormone or a chemical messenger. Makes up your hair, skin, cell membranes, muscles. So hemoglobin carries oxygen around in your blood, and it's also antibodies in the immune system. And why is that relevant? Because amino acids make up proteins. You'll hear words in the upcoming unit called like monomers, polymers. So monomers are the individual units that make up the polymers. With proteins, we're talking about the amino acids being the monomers, the proteins being the polymers. Plastic is ethene is the monomers. We join them together to make polyethylene or plastic. So polymers, long chains of small units called monomers. So amino acids. So there's 20 of them. You are not expected to know what they are. Just let me check. I'm 99% sure they're in the formula book. If you've got it there, have a look. So you should get the main ones and what they look like. My internet decides to load. So if we go down, if you're looking at your formula book now, by now you should have it printed out. By now you should be realizing what you need to know. So if you go to page 12, oh, that's Poppy. Hi Poppy, we're doing amino acids and proteins. Then we'll take a quick break and we'll do enzymes. Should take about an hour and 15 minutes for all three. Then we'll do a bunch of questions on proteins and enzymes. So I'm doing an extra bit of time to make up for last week. So if I was just saying, by now you should have your formula book for chemistry pretty well memorized. You know exactly what you need to know and what you don't need to know. So if I go to my formula book, I'm going to stop sharing and I'll bring it across here. You're given the common amino acids, as well as their isoelectric point. Again, something we'll go through later on. But you're given the 20 common amino acids, 20 there, yeah. So by now you should have this printed and be referring to it in class as well. Some carbohydrates there. It has everything you need to know to answer the question. All right, back to amino acids. Okay, so there's 20 of them, and they all have, and I'm going to give you time to write this stuff down. So I'll speak for a little bit, give you a chance to write it down, and we can go through it again. So 20 different amino acids. They are all exactly the same, except for one part, or what they call the R group. Now, we know that all the organic compounds have a functional group that makes them different. The R group on the amino acid is what makes the amino acid different to the next. They all have an amino group, they all have a carboxyl group, and they all have a hydrogen. And I'll explain what is meant by alpha carbon. I'll give you 20 seconds to write that down. Okay, so now we can visualize it. So the alpha carbon in the middle is called the alpha carbon because it has 4 different attachments. We've got the carboxyl group of our carboxylic acid on the right-hand side. We have our R group, which is like our functional group for the amino acids. And then we have the amine group on this side, and our little rogue hydrogen. So this is called the amino terminus, and this is called the carboxyl, or the C terminus, or the N. The proteins always start at the amine, and they finish at the right-hand side with a carboxyl group. I'll give you just a couple seconds to copy that down. They're called 2 amino acids because the R group comes off the second carbon. So on the left-hand side, we have the amino group. On the right-hand side, we have the carboxyl group. And when they join together to start making the proteins, the amino group attaches itself to the carboxyl group. And just remember that. So here are the amino acids. Go away, you can go there. So you can see they start off as simple as alanine with just a methyl group attached. Then you've got arginine, arginine, whatever you want to call it. You've got asparagine, aspartic acid. It's got another carboxylic acid group attached. So they are represented by the 3-letter symbols. They all have the same base structure. So you don't need to know all of these amino acids. You're given them in your final exam. I think that's overly nice. If you go to uni and study chemistry or biology or biotech, you'd be expected to know all 20 of these. So we can't manufacture amino acids. Only plants can do that. So small animals eat plants, and then as we go up the food chain, so condensation. I want everyone to think, take 1 minute to think, what does condensation? What could condensation possibly relate to? And think about what's on the end of those molecules. Condensation reaction. We have the carboxyl group of one amino acid, the NH3 group of the other amino acid. We get a reaction that produces a peptide bond and a molecule of water. Carboxylic acid and an amine react to form an amide. So we have NH2 and OH. Look what we've got. We've got 2H and an O. The HOH breaks away as a water molecule, hence the name, condensation reaction. And we get an amide, whatever you want to call it, form here. So we've got carbon, double bound to oxygen, bound to nitrogen, hydrogen, and hydrogen. So I'll give everyone 30 seconds to take that down. Oops, sorry. So you can see that here. We have glycine, which is just, its R group is just a hydrogen. Alanine, where its R group is just methyl group. They combine, and we start to build the protein chain. Now, big thing in grade 12 is you're expected to use scientific language. So, 2 amino acids combine in a condensation reaction to form a dipeptide through a peptide linkage or a peptide bond. So get used to this kind of language. Now we're out of the realms of just learning the theory behind everything. We'll do about 8 questions at the end of enzymes, which should bring all of this stuff together. So peptides. Peptides are small chains of amino acids linked together. 2 amino acids called a dipeptide, fairly self-explanatory. Many is called a polypeptide. So here we've got alanine, glycine, 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 cysteine, valine, and lysine all joined together. So here we've got the peptide bond. Here we've got the R group. So you've got to be able to recognize what's what. Here we've got the amino terminus. Here we've got the carboxyl terminus or the C terminus. And each time one of these bonds is created, a water molecule is released. So that process just repeats. Okay, so a polymer is a long chain of monomers. The amino acids are the monomers that make up the polymer here. In this case, it's a biopolymer or a protein. Okay. So this is insulin. It's made up of 51 different amino acids. So it's a biopolymer made up of 51 amino acids. Proteins can get up to thousands of amino acids. Just depends on what they are, really. Okay. Any questions on amino acids? It's fairly self-explanatory. It's fairly simple, not overly complicated. So I'm going to do a review of behind me is my uni organic chemistry students work. All right. So I want everyone to rehash the intermolecular forces and what they are, what causes them, because we're going to review them. All right. Super important when we're talking about proteins because their molecular structure is very, very important. So we have dispersion forces, which are non-polar bonds. You will be assessed on this in your exam. So there will be a question on intermolecular forces, probably even just a multiple choice one, but it's something that you really need to understand. So non-polar bonds, carbon to hydrogen, very weak, and fleeting. result from random polarization. We have dipole-dipole. When we have polar bonds. So this is when we have carbon double bound to oxygen. Partly because oxygen is more electronegative, partly because of the presence of the non-bonding pair on oxygen. Negative end, positive end, another carbon comes along. Bonding between those two bonds. Hydrogen bonds. When we have hydrogen bound to oxygen, nitrogen, and fluorine. So QCA are not going to be nice to you. They never are. I'm sure you probably realize that by now. Most of you did grade 11 last year. How much misery they put you guys through. So they're not going to ask you simple questions like, which has the highest boiling point? They're going to ask you about the intermolecular forces. Why those molecules with hydrogen bonding have stronger intermolecular forces. So it's partly because of electronegativity. And partly because of the non-bonding pairs of electrons. So in dipole-dipole forces, oxygen has one pair. In here, oxygen has 3 pairs. There's 3 pairs of extra electrons, and then it steals the electrons from hydrogen as well, because it's very green. So, strongest, middle strongest, weakest. Why is this important? Because proteins, because of, if we remember the R groups on the amino acids, they're like a tail. They have all of these kinds of bonding. So proteins will fold and contort and twist because of these kinds of bonds. That's my rant about intermolecular force stuff. Let's look at proteins. Oh no, I grabbed the wrong chair. Structure of proteins. So anyone want to have a go at what protein is? Or have a think about what a protein is? So have a think. What's a protein? Right. So proteins differ in the number, the type, and the sequence of their amino acids. Why is this important? Well, each of the amino acids has a different R group. Each of these different R groups will have different intermolecular forces present. And the intermolecular forces that are present will determine the 3D shape of the protein. And the role of this protein depends on its shape, and its shape depends on its role. You'll get to know weird looking pictures like this. So, myoglobin. Everyone remembers from PE about their ATP energy system, which gives you the ability to run full tilt for about 10 seconds. Because you have about 10 seconds worth of oxygen bound to myoglobin in your muscles. Now that looks really complicated. It kind of is. But all those twists and turns and sheets and helices and spiderweb looking things are parts of the protein that have twisted and contoured because of the intermolecular forces that are present. So we have primary, and everyone, if you've got a page there, just draw like 4 headings, split your page into 4. So my research for far too long centered around the structure of these proteins and creating pictures like that squiggly line drawing of myoglobin. It wasn't very exciting. Okay. The primary structure is the boring bit. It's just the amino acids written left to right. So we start with the N-terminus on the left. We go to the C-terminus on the right. Primary structure of the protein doesn't really tell us much. So on the end, we've got our amino terminus. Glycine is on the N-terminal, or the amine functional group. It doesn't have a linkage. On the right-hand side, we've got the C-terminal, or the carboxyl group. Remember, each time we stick 2 amino acids together in a condensation reaction, we produce water as a byproduct. Okay, so polypeptides are up to 50 amino acids, and proteins start at 50. All of the amino acids are linked together with peptide bonds. So if you're drawing or writing stuff down, the amino acids are linked left to right using those 3-digit codes, which I'll give you 20 seconds to take down. So on the underside or top side, it doesn't really matter. You would have the amino acids, and then you've got the R groups coming off the end. Remember also, on every amino acid, we also have carbon to oxygen double bond. So there's going to be some dipole-dipole bonding happening. And then you've also got, so here we've got dispersion forces because we've got hydrocarbon. Here, SH, that's sulfur, it has dipole-dipole bonds, but you don't learn about those. Really. Here, we're going to have hydrogen bonds because we've got carboxylic acid. Here, we've got amine and amide groups. So we are going to have hydrogen bonding occurring. So this guy's going to stick to the carboxylic acid. The sulfur groups are going to stick to each other, and you can see how it can start to fold because the R groups will interact with each other. That was my beautiful interpretation of a protein folding. So the entire shape of the protein determines the structure of the sequence of the amino acids determines the structure of the protein chain, of the whole protein. And they can make some weird shapes. So theoretically, we should be able to take the primary structure of a protein and figure out what it looks like, right? Wrong. There are so many interactions in a protein that the fastest supercomputers and the most powerful computers in the world cannot accurately predict the 3D shape of a protein from this primary structure. And we can sequence the proteins by protein sequencing. You don't learn anything about protein sequencing. So see, the first thing here is the sulfur groups will create bridges between the proteins. Once that happens, all of these R groups are going to start interacting with each other. Phenylalanine will interact with glycine at the top here. That will then change the interactions all the way down until eventually it settles in its shape. Now, like most things in chemistry, it will settle in the shape that requires the least amount of energy to maintain that shape, if that makes sense. So, bovine insulin is from pigs, I think, bovine, yeah. What's that porcine? Anyway, they used to take insulin out of, so well, before I do that actually, insulin is used to treat type 1 diabetes. People with type 1 diabetes cannot create their own insulin. They cannot regulate their own blood sugar levels. There is a 3 amino acid difference between human, pig, and cow insulin. The latest and greatest craze 50 years ago for insulin was to take the insulin out of the pancreas of cows that were slaughtered, pigs that were slaughtered for food, purify it and inject it into humans. And it worked. But it only worked for a year or two. So unfortunately, what happened is because it was ever so slightly different by 3 amino acids, even though that doesn't sound like much at all, the body knew that it wasn't human. And eventually, it learned how to just destroy it as soon as it was injected. So the primary structure has a massive flow-on effect to the other structures. So now we're getting to the real stuff. So the secondary structure folds the protein like this. So we know that the amine groups have hydrogen bonding. which can interact with the carbon to oxygen double bond on the end of the carboxylic acid. So you've got a straight line, and now because of the interactions, it's starting to twist into a never-ending S. Because of this hydrogen bonding between the amine and the carboxylic acid. So, NH is positive, CO is negative. Now look, NHCO, NHCO, NHCO. So this isn't anything to do with the R groups yet. So, because of this, we have alpha helices and beta pleated sheets being created. So, alpha helices are caused by this H plus bonding with the negative oxygen. So twists itself into a ribbon shape because energetically that is the most favorable position. Those hydrogen bonds will physically force the straight line to begin to twist up into this helix shape. Same reason the DNA is the double helix. So beta pleated sheets is the same thing, but when we get multiple protein chains aligned with each other, and it creates it's not really a corrugated ion effect, is it? A pointy corrugated ion effect because of the hydrogen bonding between oxygen and hydrogen. If you can hear a dog whinging, it's because she's being crate trained. She does not like it. Okay. So if the long polypeptide chains fold over themselves, because again, it's never going to sit in a straight line because you've got all of this energy there in those bonds that needs to be released or used. Release is probably not the right word. The tertiary structure is the overall 3D shape. Now, this is where the R groups come into play. So they will fold up into the alpha helices and the beta pleated sheets, depending on the amino acids that are there, purely because you've got NH and CO, and they will naturally go into those. Now, the difference in the overall 3D shape comes from the binding between the R groups in those secondary structures. And this is why we just cannot go from the primary structure to the full tertiary structure. There are so many possible combinations that it's impossible with our current technology. So now the R groups, some of them are long, some of them are more reactive than others. And for lack of a better term, flapping about, and they can then interact with each other and further twist and contort the protein into its 3D shape. So, oh, that was wrong there. Remember these guys? So, the side chain, depending on what structures are there, will either have hydrogen, dipole-dipole, or dispersion forces. She is not like that. We can also have ionic interaction. So positively and negatively charged ions will interact with each other. So amino acids called cysteine has sulfur in it, or a sulfide bond. So when you get one on one side of the protein chain, another cysteine down here, they will link together to create this disulfide bridge. Very important structure in protein. Sulfur is an essential micronutrient. If you don't get enough sulfur, you can't make proteins, your body doesn't function properly. You can see the disulfide bond there, linking the two parts of the polypeptide chain through this covalent bond between sulfur. Covalent bond, sulfur is sharing its outer electrons. So if you ever see SS and they ask you what it is, sulfur and sulfur. So this is why it's so important, right? So you talk about 3 amino acids different. Certain amino acids you can change and it won't affect anything really. I might change it a little bit, but not enough to really notice. But if I was to add in 3 more cysteines, and I'm pretty sure human insulin only has 2, it completely changes the amino acid. I mean the protein. Even though it's only a very small number of amino acids that we've changed. We think of tertiary structure. Primary is primary school, secondary is secondary school, tertiary is like university. So it's the 3rd structure, the 3D structure of the protein. And the R groups will interfere with the bonding, as well as the alpha helices and beta pleated sheets. Now there is one more structure, but the important definition to make here is that this only occurs when there's multiple protein chains, or the quaternary structure, or quarter 1/4, it's the 4th level of protein structure. In case you didn't know how many numbers 4 was, so it is when we have multiple protein strands, like insulin is one of them. We also have, I'm pretty sure the example is here was hemoglobin. So red blood cells carry hemoglobin. Hemoglobin carries oxygen, and it's made up of these 4 different polypeptide chains. These little bits here, they're called ligands. I don't think you need to know that for senior chem. But what happens is the oxygen binds to these spots. So 1 hemoglobin molecule carries 4 molecules of oxygen. And again, you alter. So if you're doing biology, you should know what sickle cell anemia is by now. And it's a very slight change in the primary structure of hemoglobin, but it makes it 50% less effective at carrying oxygen. Also changes the shape of the red blood cells, but I won't go into biology because it's boring. Okay, so primary structure is the sequence of amino acids in the polypeptide chain. Secondary structure is the alpha helices and the beta pleated sheets. And that's caused by the hydrogen bonding between the NH group and the CO group of the carboxylic acid and the amine. Tertiary, now we're starting to get the shape affected by the hydrogen bonds. So we've got hydrogen bonding occurring, we've got dispersion forces occurring. We can also have dipole-dipole forces. We've got covalent bonding between sulfur. We've got ionic bonding between the negatively and the positively charged species. And now it's pulling itself into the tertiary shape. So, quaternary is how do all those tertiary structures come together to make up the overall shape of the protein. Oh man, that was a lot to get through in 2 minutes. Okay, right, we're gonna do 10 minutes. Right. Don't worry about question 1. So I'm gonna give everyone 2 minutes to answer question number 2. All right, 1 minute. All right, yeah. Okay, so the secondary structure represented here is the alpha helices of alpha helix, and it's formed by the NH formed by the hydrogen bonding between the CO and the NH2. So I'm going to run another one after this if you guys want to join back in to make up for last week on enzymes. Probably another half an hour or so on top of this. But I'm going to upload the recording. I'm going to send out this set of slides so that you guys can answer the questions as your homework, and then we'll go through the questions. There's also questions on enzymes as well. The first 20 minutes of next week, we'll go through these answers. Okay, so I'll stop the recording.